All right, first order of business. Annie, remind me to mark you here. I'm not going to do it right now because I'm recording, but remind me at the break. So first order of business is the final exam schedule. Remember I complained about it at the beginning, and I know what it looked like in the beginning because those of you guys in my lab, I printed it out for you, right? And this is what was posted online in the student handbook at the beginning of the semester. Somebody went in and changed it, which is somewhat good and somewhat bad because they still managed to screw it up. So when you look at it now, right, the good news for us is we're on Monday night, right, for our exam, except it's at 8:30 at night. Oh, wait a minute. Was I miss? No, sorry. Tuesday. We are on Thursday night, Tuesday, Thursday classes, instead of on Monday, or on Thursday, right? Except that's definitely at 8:30 because the math exams are here. So the other problem, too, is that these two used to be up here, but on switch days, the Monday, Wednesday, and Tuesday, Thursdays, and some idiot put it down here, and I don't care who it is, I'm calling them an idiot. Put regular class exams in with math exams, so we have a conflict. So I've notified my boss, and I've notified the head of the math department that this is what the current schedule says online. Okay, so that's where we are at right now. So the other thing I asked my boss was, okay, I'm thinking my students probably don't want to take the test Thursday night at 8:30 at night. Can I give it in this slot right here on Tuesday night at 6 o'clock? And he said yes, if everybody agrees. So we're going to vote. Several people who were here earlier already heard this discussion and have already voted. Does anybody want to do it Thursday night at 8:30? Thursday the 21st. Yeah, so this totally screws your schedule, Annie. So no matter what, I don't know, it might actually make it better for you, but no matter what, we can work around things too. Okay? Yeah, so our lab, right, your lab, you're the only one that's in my lab, will be Monday night at 6 p.m. Okay? And then this one will probably be, I'm thinking, Tuesday night at 6 p.m. So I don't know what your other classes is that you have, when they'll actually be.
Oh, no, that's when.
Yeah, well, we're going to do it anyways. Yeah, okay. The 26th at 6 p.m. Okay? So the only people not here voting, let me update my attendance. I'll go ahead and do it right now. I don't care. It'll go on my recording, if I can find it. Attendance. And so I will change that in here to in Canvas, so it'll reflect that. So Aubrey came in, Audrey with a D, and Annie. So Ashley, Janice, Brittany, and Makisha are not here. But those four will probably fall in line with us. We'll see. Okay. So once I check in with them, I'll make it official. But it looks like I don't see why anybody would pick that. But, you know, you don't know. But I could always make other arrangements too. Okay. All right, so that takes care of that. That leaves us, if you actually look at the calendar, oh man, I am going to have to plug this thing in. Okay. Tonight and Thursday night is it. That's it. Because the 19th, you guys will take test 3, right, on this stuff. So that leaves us not a lot of time to finish what we have left. So one of the other things that I want to change, propose a change to help you guys, is that you see this bonus assignment? I had originally put it due for, let me edit, for the day of the exam. I'm going to move that up, but you guys don't necessarily have to submit it. But I want you to watch these 4 videos and take notes. You can type up notes. You can handwrite notes. You don't necessarily have to submit it, but bring it to class with you Thursday. Make sense? Right? So basically, I want proof that you watch these videos, right? Write something down or type something up. For this Thursday, right? So I'm changing it right now to Thursday at class time, 6 p.m. Yeah, oh, sorry. Wrong one. Thank you. All right, so saving, updating. That's step 1. Okay.
Thursday, there's a barista competition going on, and it's all day, and I'm part of it.
Mm-hmm.
And I read it. It says I can take a picture of my notes and load it up.
Yeah, you can do that too.
On Wednesday, I can just send it to
Yeah,
you.
yeah. I mean, if you guys want to submit, you can. Like, you can just submit it on there. It'll make it easier for me grading. But you can also just show me in class, and I'll, mark you as completed. So, either or is fine. Right? But, since I'm going to actually see you guys, I'm not going to make you take pictures of your notes, But if you want to, right, you're a techie person, go for it. But definitely, before class on Thursday, watch these videos. They're not too long. They're my YouTube videos. It's an activity that I did in class, and I was hoping to do a version of that with you guys, not realizing we're kind of don't have time. But I will say this. If you guys want, Tuesday before the test, we could have a powwow session and talk about stuff. Maybe do a little activity or something before you take the test. Depending on how you guys feel. If you guys feel good and you're like, shut up, Erica, let's just take the freaking test, I'm fine with that too, right? Because, as soon as we get done the test or whatever we do that night, that's it for that night. And then we don't meet back, if we all agree, until the following Tuesday, the 26th. And you guys will do your final then. So, yeah. Okay. I felt like there was something else I'm forgetting. I don't know. Okay, so when I was looking at Module 3, I was noticing this is still the student version. That's not a complete version of that. So I'll fix that during break, where it has the version I'm going to show you guys. So remember last time we didn't finish up cells, so we need to finish that. And then we're going to move on to first line defense, which is this lecture. And you guys are going to learn about antibodies and stuff like that by watching those animations and stuff. So I might skip to adaptive part B after break. So if some of you guys like to print, haven't printed yet, maybe during break you might want to go upstairs and print that one. I don't know, my print people here tonight? Who would like to print? Okay, no. Okay. All right, so that's the plan. That's the plan of attack for tonight. Sound good? Okay. All right, so let's rock and roll. So let's go back to cells. What's today? Today's Tuesday. I wanted to bring those plates over from lab today, and I forgot. Maybe I'll run over to the other building during break so you guys can see them that aren't in lab. Who's not in lab? Dylan. Christy, you in lab? Okay. Alright, maybe I'll do it Thursday then, since Dylan's the only one who hasn't seen them. Away from more peeps that aren't in lab. Okay, what am I looking for? I'm looking for lectures. Alright, so we're going to do the cells, right? And I need this version. And we were just on communication for that one, right? We only had not too many slides left. And then that brings us to innate response, which that one isn't too bad. We should be able to get through. Okay. And then I gotta post that there during break. All right, see here, this is where I was changing, because again, I still don't know for hers, for Sarah's, if she's actually going to do it on Thursday or that Tuesday. So, I don't know. We'll see. Yeah, I know. That's where they moved it to, but it's conflicting with math. So we got to figure out what math is going to want us to do. I mean, we could probably even move it up like I have here to 1 o'clock. Right? But we can't give exams during math, especially if students have conflicts. Like if they have math takes precedence usually over us. She does. I talked to her. I talked to her after you guys' lab. So she knows that I was coming right over here to talk to my boss, which I did, but the math person was gone. So I'm waiting to hear back before we make a final change to the schedule. Uh-oh, which one is my cells? The cells. Here we go. The cells. All right, so I think we're okay on the cells, right? Let's hope. So we're on cell communication now. I think this is right where we ended, right? Yeah, okay. All right. Okay, water. Oh, that was the other thing I noticed. This thing looked like it was getting low on power. I don't want it to shut down in the middle of me talking. All right, so list the functions of the following molecules. So surface receptors, right? What do these do, right? Why are they there? Cytokines we've been already kind of talking about, right? So we're going to go into a little bit more detail. And we've already seen adhesion molecules in action, right? During inflammation. So, and their name themselves pretty much tells you their job, right? To stick to stuff. Adhesion, right? Grab onto, stick to stuff, right? Which is very important. So some of the, some terminology that would make more common knowledge to us. If you think of surface receptors, so these are receptors on the surface of cells, but cells also have receptors on the inside too, right? These allow them to sense their environment, right? So they are essentially their eyes and ears, right? Allows them to see and hear what's around. They don't truly see or hear, right? It's all about molecular interactions. In this case, those molecules interacting with the receptors. How messages travel for them is via cytokines. So similar to my voice, right, that's being picked up by your ears, those receptors, right, cells send out those signals, those chemical signals we know as cytokines, and they're received by the receptors, the aka ears of the cells. Adhesion molecules, as the word implies, right, allow them to adhere. So very, again, similar to our own hands, they actually are meant to be able to grab onto things. And we've already seen these in action in inflammation, right? They're very important for guiding those white blood cells to the area where they're needed. So surface receptors, these are proteins basically, embedded in membranes. And they send this signal, this information inside the cell. So those proteins in our membrane, some of them, right, allow stuff to go in and out of cells. They're facilitative proteins, help with facilitated or active diffusion. But a lot of them are there as receptors to be able to sense the environment, see and hear what's going on outside that cell. So there's some terminology, right? Receptors that are specific to the molecule to which they bind. The molecules they bind are called ligands. When a ligand binds its receptor, it becomes modified and sends a signal inside the cell. And of course, the cell is going to respond by initiating some action, right? So if I yell fire, what are we all going to do? Jump up and run out the building, right? So that's a response, right? For our cells, if there's a particular trigger, right, they're going to respond in a particular way, always, because they're programmed to respond in that way. So probably, again, remember, cytokines and hormones are similar, right? And that they are basically signals that tell one cell from another cell what to do. So one of the most common hormones that most people are aware of is insulin, right? Insulin binds to your cells and basically tells your cells, hey, there's glucose in the blood. Go ahead and eat it, right? Diabetics run into problems because they don't produce insulin, or the insulin they produce is ineffective. And so their cells don't know when glucose is present in the blood. And so they're not able to take it up into the cells, right? They can't sense that it's there, right? They don't have receptors for glucose. The insulin that binds to the receptors on the cells tells the cell glucose is here, right? You can eat that. So if they can't eat it, then they eat other things like proteins and fats, and they produce acidic products, and that can be really bad problem, right? You can end up in ketoacidosis. You can die from that. So cell signaling is very important, right? So insulin's a very good example of, if you don't have that signal, you're going to have problems. Same is true for our cells, right? Except this is a little bit more complicated communication, right? You think hormones are bad. Cytokines is a whole nother monster, right? So we'll look at a couple of examples of actual cytokines. So as you can imagine, as I said, there's a lot of them. But there are ones that have similar interactions, or you could say functions. So there's a whole group of them that are lumped in and referred to as chemokines, because these are involved in chemotaxis. So chemo means chemical, right? Because, again, these are chemicals released by our cells. But in this case, when it binds to another cell, it tells that cell what? What does it tell that cell? So if I release a chemokine, and what is chemotaxis? What are they going to do? What's taxis? Why do we take a taxi cab? To get somewhere, right? Chemotaxis is movement, taxis, movement, based on a chemical signal. So when we have inflammation, right, they send out chemotaxic cytokines that say basically, come here. Right? Come right here, I need help now. And they're specific as to who they call, right? One of the ones that we're going to look at tonight is a complement protein called C5a. It specifically says to neutrophils, hey, you, come over here. I need your help. Colony stimulating factor we already talked about, right? So this colony stimulating factors, there's multiple ones, right? There are specific ones that will travel from places in our body to where are all our blood cells born. From hematopoietic stem cells found where in your body? In your bone marrow. In your bone marrow, right? So that signal is going to go straight to the bone marrow and say, hey, we need neutrophils. We've got serious bacterial infection right now. You guys need to make a whole bunch more neutrophils. Right? That'll be that specific colony stimulating factor, right, will cause those hematopoietic stem cells to go down that lineage. Or it might be an all hands on deck, right? Several colony stimulating factors, like every white blood cell you can make, just make it. Right? We've got a serious problem. We need everybody. There's that word again, leukocytes. What does that mean? What's the common term for a leukocyte? White blood cells. All of them, right? All the white blood cells. So interleukins, and I know I skipped one. Interleukins. Inter means, right? If we take an interstate, we're going between states, right? Interstate, right? Traveling, right? So interleukins, leukins referring to white blood cells. So these are white blood cells talking to each other. And some of them even will talk to themselves. They're kind of like me, like I talk to my phone, even when Siri isn't on. Some of us are more verbal, right? You know, when I'm alone in a room, and I think I'm alone, I'll sometimes verbally answer a text before I actually type the text. This can be confusing when someone walks in, they think I'm talking to them. Our white blood cells do that very same thing. Some of them will release interleukin-2, which will then bind to their very own receptors and tell them to proliferate, as well as neighboring cells. But they literally will tell themselves what to do. I kid you not. Right? This is an autocrine function. Some cells of the immune system, some cells of the endocrine system do stuff like that. But it's more common, definitely, in the immune system, for them to literally talk to themselves. So there's a ton of interleukins, and so they're abbreviated IL, and then they're given a number designation, probably in the order in which they were discovered. So I don't even know what IL we're up to. But as you can imagine, IL-1 and like IL-2, you see those a lot. And so it makes sense that they were probably some of the first discovered, right? Because these cells use that particular signal a lot. And so we'll look at some specific examples, but I just want you guys to know the generalities, right, that's covered in this slide. Although for some of us, having specific examples, right, helps us understand what we're talking about. Interferons, so interferon, we're interfering specifically with viral infection. So this is a signal and a signaling pathway we'll go into detail with for interferons. This one we will cover in more detail a little bit later. Tumor necrosis factors, again, as the name implies, we discovered these when we found that actually cells will release this and will actually have caused tumors to die off. And so, of course, they've used this in therapies as well as interferon. Sometimes when you watch commercials and stuff on TV, they'll talk about certain antivirals, and they'll actually mention this one does not contain interferon, right? Because some of the antivirals actually give you additional interferon, right, to boost to get even more in your system than what your cells are producing, to let the system know what's going on. So here are some specific examples. So as I said, the interleukins are commonly abbreviated IL, and then they're given a number designation. So as an example, interleukin-1 can be released by macrophages and epithelial cells. It will bind to T cells and activate them. It can bind to macrophages and activate them. So again, they can talk to themselves and to neighboring macrophages. It also can start the fever response, induce fever. IL-2, as I said, released by T cells and causes T cells to proliferate. So the T cell that releases it will actually even cause itself to proliferate. So again, by no means memorizing these individual ones, but just give you an example of specific interactions. And some of the videos, animations that I have you guys look at, they actually give specifics, right? They'll say IL, right? Whenever you hear IL, you know that's interleukins, that's a cytokine. That's a chemical message from one cell to another or even to itself. So adhesion molecules, as we said, we've already seen these guys in action, right? They're responsible for recruiting those phagocytes to the area of injury. I just realized I left my mail open, so it's going to ding at me, so I'm going to turn that off. So we don't have to listen to that. So these are pretty self-explanatory, right? So the next we're going to look at is a little bit more detailed receptor engagement and protection systems, sensory systems. And again, we really want to focus on what do these systems allow us to do? What are the outcomes? What happens when these guys are triggered? So what happens when Toll-like receptors are engaged? Right? What happens when complement system is activated? What happens when, right, we detect long double-stranded RNA? Which right there, double-stranded RNA. Do we have that? Double-stranded RNA? No. What microbes could potentially have double-stranded RNA? They can make us sick. Most scientists don't consider them living. Viruses. Right? So this is, again, a specific interaction for us to be able to detect viral infection with double-stranded RNA viruses. But as we know, viruses come in all different nucleic acid combinations, right? You can have single-stranded RNA, you can have single-stranded DNA, double-stranded DNA, and of course double-stranded RNA. And if it's single-stranded RNA, it could be plus or minus, right? That's a whole nother ball of wax when it comes to the virus itself replicating. But again, we modify our messenger RNA. So ours, even though it's single-stranded, looks different than viral single-stranded RNA even. So we have ways of detecting that there's foreign nucleic acid in us, potentially from a virus. But we have a specific receptor for the double-stranded RNA. So these systems are within the blood, and they can detect signs of tissue damage or microbial invasion. And they respond to patterns associated with danger. All right, so coming up, there's a whole bunch of acronyms. We have way too many acronyms in immunology. You get to the point where you stop forgetting what start remembering what they stand for. Some of these can directly destroy the invading microbe. Others can be a signaling mechanism to recruit other defenses. The important thing is that they're a censoring system. Right? They're going to let us know that invasions happen. And how they know this is these patterns, right? These molecular patterns. So they're pattern recognition receptors, or pathogen-associated molecular patterns. Basically, they're highly common molecules as it applies to the things that make us sick. So it's made sense that those receptors have been encoded in our DNA and have been passed down generation after generation after generation. They've been conserved, right, because they've been beneficial to us. So having a receptor that can specifically detect cell wall components like peptidoglycan, right? We know bacteria have peptidoglycan. So if we have a receptor that detects peptidoglycan, that's going to let us know, oh, bacteria got where he wasn't supposed to get. We need to let the system know this is bad news. Now for lipopolysaccharide, what type of bacteria have that? Gram-positive or gram-negative? Gram-negative. It's part of that outer membrane, right? The outer leaflet of the outer membrane. And that's really bad news for us, unfortunately, because when it breaks apart into its different components, it's toxic to us. So sometimes even when we kill off gram-negative bacteria, we're actually making it worse for ourselves because the components of their membrane, their outer membrane, is toxic. That's why you have to be really careful with gram-negative infections, especially when they get to the bloodstream. right? Because we can't just give you a whole bunch of antibiotics because we're going to kill a whole bunch of bacteria really fast. And then we're going to flood your system with this endotoxin, with this lipid A gram-negative endotoxin, which, as the name implies, it's toxic. So we just made the situation worse. Teichoic acid. Who has that? The teichoic acid. Nope. Gram-positive or gram-negative?
Gram-positive.
Gram-positive. It's like the rebar, remember, amongst the many layers of peptidoglycan. Lipoproteins, right, that are unique to microbes. This could be both gram-positive and gram-negative. Flagellin subunits, the protein that makes up the flagella of bacteria, is unique to them, right? We have receptors that can detect that. And then we have ones that can detect viral RNAs. So again, they're given multiple names. The important thing is, right, whether we're talking about microbe-associated molecular patterns or danger-associated molecular, we're looking at patterns, right, that are repetitive amongst the things that hurt us, that can harm us, bacteria, viruses. All right, so quick video.
Structures on the surfaces of phagocytes, called pattern recognition receptors, make it possible for the phagocyte to immediately recognize a number of different components of infectious agents, referred to as pathogen-associated microbial patterns. Toll-like receptors are a class of pattern recognition receptors that play a critical role in the early innate immune response to invading pathogens. Several different toll-like receptors located in the cell membrane or endosomal membranes of phagocytic cells have been identified. They recognize lipoproteins and lipoteichoic acids, viral double-stranded RNA, lipopolysaccharide, and bacterial flagellin. Some toll-like receptors.
So notice the one that recognizes double-stranded RNA. Where's that receptor? It's T3. TLR3. It's not in the membrane, is it? It's on the inside in an endosomal membrane. Why is that receptor on the inside? Why isn't it on the cell membrane?
The viruses release the RNA into the...
Right. Remember, the viruses fuse with our cells and gain entry inside. So that RNA is remember, surrounded in a protein coat and maybe an envelope, especially for animal viruses like ones that infect us. That RNA is not on the outside of that virus. So when that virus comes bumping up against this cell, there's no sense to have a receptor there. It's when the virus gets inside of our cells that then the RNA is going to become part of the inside of our cells. It's become visible to our cells, right? Because it's going to come out of that protein coat, out of that envelope. So then it can start being copied by our cells. But that's when our cell receptors can recognize it and go, oh no, right? Bad guy is here. So again, what we want to focus in on is, okay, we can detect these things, but what happens next? Okay? So think about it, right? If I yell fire, what are we going to do? We're going to run out the building, I hope, right? Yes, maybe so. I know we're tired, right? But really, if the alarm goes off, we're leaving, right? Or, if I see a fire, we're leaving. What do you think this cell's going to do? Right? It's a phagocytic cell, they said, right? It's detected maybe, let's say, lipopolysaccharide. It detects lipopolysaccharide. What's it going to do? It's going to eat it, right? A phagocytic cell is going to phagocytize it, right? And then it may even send out additional signals telling the neighboring cells, right, we've got invasion here. We need backup, right? They may call in their neutrophil buddies. So how is it they're going to be able to do that? Right? How does a cell know how to do anything it does? It's encoded in their DNA. So watch what happens.
Receptors exist as monomers, whereas others exist as dimers. When a Toll-like receptor interacts with a particular pathogen component, it activates specific cytoplasmic adaptive proteins such as MYD88, MAL, TRIF, and TRAM. This activation initiates a signaling pathway to defend against a specific type of pathogen. Lipopolysaccharide is a pathogen-associated structure produced by gram-negative bacteria. It first binds to lipopolysaccharide binding protein and then associates with a cell surface protein CD14. The CD14 with bound lipopolysaccharide binding protein then activates TLR4. The activated TLR4 then interacts with and activates specific cytoplasmic proteins. The interaction of TLR4 with these cytoplasmic proteins activates other proteins, allowing them to convert inactive transcriptional activators such as NF-kappa B into active forms. The activated NF-kappa B moves into the nucleus and binds to specific sites on DNA. Binding of NF-kappa B to specific sites on DNA induces transcription of genes for cytokines, including interleukins and tumor necrosis factor. These cytokines move out of the phagocyte and stimulate various aspects of both specific and nonspecific immunity.
So, the unfortunate thing is they go into quite a bit of detail about what molecules are actually involved in the cytoplasm, right? And yet again, a multitude of acronyms, so then you start to want to fall asleep. But remember the big picture here, right? The signal is sent all the way to the nucleus, right? That's where we know how to do stuff, where it's encoded, and then it's going to be determined, okay, release these cytokines, right, in response to this stimuli. Make sense? Right, so we don't have to, get bogged down by the details. Right, it's the overall picture we're interested in. So Toll-like receptors, as they said, are a group of these pattern recognition receptors found on the surface as well as inside of our cells. Right, and so it makes sense that sentinel cells like macrophages, right, that are always on guard in our tissues throughout our body, that they would have these types of receptors, and they would be able to respond directly, right? They can directly destroy, but they can also call in reinforcements. They can call in help. Dendritic cells can do the same thing. Cells lining sterile body sites, right? So areas where pathogens bacteria, nothing should be getting in, right? Those cells have these receptors. They can say, oh no, right? Something bad is happening. They can send out the signal and get help. They themselves can't necessarily eliminate the problem, right? They're not phagocytic cells, but they can call in phagocytic cells.
Is there the same amount of these receptors in all of those? Or is there, like, how there was, like, the T4, T whatever?
Right. That is a very specific question that I don't know the answer to. But I could surmise that it would make sense that, that they would pretty much have, so she asked, like, how many? So you're, like, saying how many Toll-like receptor number 1 is on each one of these cells?
Right, each one of the different types, like the macrophages with the dendrites and the location.
Right, yeah. I don't even know if researchers have done that level of research, to tell you the honest truth. But, it would make sense that, they would have a good enough number that if they come in contact with these pathogens, they would be able to sense them, right? Yeah. So it's like imagining, us, right? We have 2 eyes. But flies, they say, have, like, how many eyes? I don't know, an insane number of eyes. So, it doesn't matter. I can never kill those suckers. Forget about it. I'm not fast enough. But, I would think that they'd have enough receptors that they would be good at sensing their environment. Does that make sense? It's like, if you have a house, you're not going to put in one of those alarm things when someone opens your window on just one window, right? That would be pretty stupid because, they could get in some of the other windows to get into your house. Does that make sense? All right, so they'd have, a significant number of these receptors on their surface, because that's one of their main jobs, is to be able to sense these, and especially the phagocytic cells, eat them, but then also signal to other cells. So remember, what's the important thing? Following detection, the signal is transmitted to the nucleus, and they're going to do something about it, right? They're going to express genetic information, right? And the most common response, and this is why we started out with it, is an inflammatory response, right? If we've got invasion, not only are these phagocytes going to gobble it up, but they're going to call other cells to that area to assist them in this invasion. And if we're dealing with specifically a viral attack, then we're going to do a viral response, which is actually different than how we respond to bacteria, because it's a different type of invader, right? We can't deal with them the same way. They don't affect us the same way. They don't do the same things to our cells. We have to combat them in a different way. So we do that, right? We respond to viruses in a manner that should eliminate them, right? Or at least hopefully stop their spread. And bacteria, we really want to get rid of them. So NOD-like receptors are found in the cytoplasm, right? Because again, think about this. Does stuff always stay outside of our cells? No, some cells, like phagocytic cells, will actually purposely take stuff in, right, and break it down. But there are intercellular bacteria, right, bacteria that actually get inside of our cells, that grow inside of our cells. Mycobacteria tuberculosis is an example of that. It actually grows within the phagolysosome, right, of a phagocyte, of a macrophage. How messed up is that, right? That's supposed to kill them. And they're like, ha ha, home. I'm good. Not good for us. So it makes sense that, if stuff can get into our cells, we need to have detection inside of our cells as well. And again, notice that these Tolllike NOD-like receptors found in the cytoplasm, they'll form an endosome, inflammasome, right? So they're going to form a particular structure that, of course, that's going to cause our cells to do the most common type of response, and that is an inflammatory response. Gets its name from that, right? Inflammasome, right? So when these guys are triggered, it's going to be an inflammation response. So notice some of these examples, right? Flagellin, peptidoglycan again, but double-stranded RNA, single-stranded RNA that's uncapped, because, as I said, we modified our RNA, right? Ours becomes capped. So viruses don't do that, so we can detect that it's viral single-stranded RNA as opposed to our own messenger RNA. And then, if there's being damage happening inside of our cells, right, we can detect that. So that's the end of that one. But as I said, I want to start this next one, which is first line, before we take our break tonight. This one isn't too long. So our first line of defense is our innate immune response, right? And this is working before you're even born, right? You have certain levels of protection. You have anatomical barriers, right, that prevent the stuff from getting in to begin with, right? So what's our actually largest organ of our body and the most protective? Our skin, right? It is actually, in fact, an organ. It's made of multiple tissues working together, right, in coordination. And then we have openings, though, right? Mouth, nose, eyes, anus, vagina, urethra. Your ears actually don't open to the outside. You have tympanic membrane. Protects you. Right? But, so, what do all these have in common, like your nose and your mouth and your vagina and your anus? They're all what? They're moist, aren't they? Because they're bathed in mucus, which is protective. Right? Same thing with your tears too, right? This is a liquidy substance, not quite as sticky as mucus, right? But it's protective. So that's an anatomical barrier as well, the mucus that we produce. And then as we've already seen, right, we've seen one group of pattern recognition receptors, right, in the cytoplasm and on the cell surface. We're going to go in specifically into interferon response in this lecture. And we're going to look at the complement system, which is also another sensor system that's found in our blood and our tissue fluids. We've already looked at phagocytic cells, right? So what are some white blood cells? Their main job is to phagocytize and destroy invaders. Who's the most powerful killing machine out there, white blood cell wise? What's his name? Can't really see his granules when he stains. His nucleus is neutrophils. Remember, suicide bombers, right? Who's the big eaters that's found all throughout our body all the time? Macrophages. What are they called in our blood? Monocytes. Good job, Christy. She's like, I'm here. Yeah. It's all right. What's another one that's a close relative to monocytes and macrophages, but he has long projections that he can grab stuff with? Dendritic cells, right? So again, when we say phagocyte, think of those cells, right? Think of neutrophils. Think of them gobbling up, right? See that video in your mind, right? Because they're, really important to our defense, because not only can they detect them, they can actually destroy the invader right on the spot. But the dendritic cells, as we'll see later, and you guys will see in the videos you're going to watch, they'll actually share this information with other cells. So will macrophages, right? That's one of the main jobs of the dendritic cells. That's why sometimes we call them the scouts, right? They go out, they gather information, they bring it back to home base. They're really important information gatherers as well as they'll destroy the invader itself. Cytokines, as we've seen, and those are very important, right? Those are the chemical messages. That's all the voices, all the talking that those cells do. And let me tell you, there's a whole lot of talking going on in the immune system. Blow your mind, right? Inflammation is what we started with, right? And is paramount to our defense, right? Because we've got to get the troops on site, right? We need to be able to call them into that area from the blood into the tissue where invasion is happening, where damage has happened. And fever is the last thing that we'll talk about. And believe it or not, it is actually a response of our system, right? But as we know, it can be bad too. So, what are the common bacterial molecules that Toll-like receptors recognize? So, which of the following is not a bacteria molecule recognized by a Toll-like receptor? Double-stranded RNA. So which white blood cells phagocytize invaders, dead cells, and debris? Right. You guys just answered this one. Don't second guess yourselves. It's all the above. Which class of cytokines tell the bone marrow hematopoietic stem cells to make more white blood cells? So this would have been a good review had we got through the other part of the lecture. Which class of cytokines tell the bone marrow hematopoietic stem cells to make more white blood cells? Yes, colony-stimulating factors. Right, think of a colony as a group of cells, and that's actually what they're referring to here, right? So think about it. There are different groups of white blood cells, so we really need to signal, right, which group do we need, or do we need them all, right? That's what colony-stimulating factors allow us to do, is to actually stimulate a particular group of white blood cells if we want to. Which class of cytokines do all white blood cells use to communicate with each other? Interleukins is what I was looking for, but in truth, that's so general of a statement that what? They all could be true. They all could be true. All of the following trigger an inflammatory response except yeah. And we haven't looked at complement system activation yet, right? But we definitely saw that engagement of pattern recognition receptors, inflammatory response can happen. Complement system, as we'll see, inflammatory response is part of that activation a lot of times. And interferon induction of antiviral protein synthesis, does not elicit an inflammatory response. This is a little bit different response. And tissue damage, as we know, right, that's one of the key indicators of inflammatory response. So when we're looking at the security of our body, right, it's not all that different from a security of a building, right? We have security walls, right? Most of us have fences around our homes, right? That's our first barrier to someone breaching. For our body, that's equivalent to our skin and mucous membranes. And those mucous membranes, that mucus, not only is it sticky, right? So imagine putting like slime in front of your gate, right? And stick the bad guys in the slime before they get to us. That's essentially what you're doing with your mucus. It's sticky for a reason. It's so that the bad guys get stuck in it, right? And it flows most of the time, right? The mucus flows out of your nose, right? We refer to that as snot, right? When we overproduce it. Why are we producing it? Because there's something in there we don't want in there. We want to push it out, right? Because it'll get trapped in that mucus and it will go where we push it, right? So you're continually producing mucus in your lungs, right, that traps all that crap that you inhale. And you're continually pushing it up with the ciliated cells in your respiratory system so that it goes down the back of your throat and you swallow it and they get destroyed in your stomach, right, by the acids in your stomach. Same thing when you get the postnasal drip, right? There's a reason why it goes in that direction. We're trying to get it out, right, down to the stomach, through the intestines. Your digestive tract is just an open, hollow tube through your entire body. It's actually an external environment. It's only if it travels from the digestive tract into the body that it truly gets in and can potentially be a problem. Right? What goes in eventually usually comes out the other end, right? This is very true if you eat corn and you don't chew it. Because what corn is an embryo, right? And look at vegetable. Cat.
Hey, it's not me.
It's not meat. But this is why it's a problem too, and why you should chew your vegetables. Because it's encased in a cell wall, right? It's a single cell, right? A kernel of corn is a single cell. Okay? What surrounds that cell? Cellulose. That makes up the cell wall, right, of plants. Do you have any enzymes to digest cellulose? Not a single one. So guess what happens if you eat a whole corn kernel and you don't bust it with your teeth? It goes straight through and comes out exactly the same the other end. You got zero nutritional value from it. Probably got your poop to move through the system though. That's what we call fiber, right? Cellulose is what we call fiber. We can't digest it. So eventually it goes out the system. So how is it that cows are chewing on that stuff all the time? No, they don't. They have a bunch of stomachs. What's going on in those stomachs? They have bacteria. They purposely have all that extra room to actually grow bacterial environments that actually they digest the cellulose. And then the cow eats the bacteria. So they purposely are growing bacteria in their guts, in their rumen, right, to then digest it. So this is why, as a veterinarian, if you go to give a cow antibiotics, Most of those farmers will freak out because if you don't give them properly and correctly, you could eliminate those microbes in that cow's gut, and what are you going to do to that cow? You're going to kill it. This is also why it stinks really bad, and also what do they do all the time? Chew and chew and chew to break down those cell walls. They actually, well, I won't. They spit it back up and chew it over and over again. I don't know that they actually regurgitate and chew, but they definitely have the system where it's the microbes that are digesting. They do not, there are no animals known to produce enzymes to digest cellulose. Only bacteria do that. Interesting, huh? All right. So the good news, though, bacteria, right? They're made of stuff like us, right? Lipopolysaccharides. Phospholipids is their membranes, just like ours, right? They got proteins on the inside. They get nucleic acid on the inside. So if we can digest them, we can use them just like a food source too, right? So if you swallow it, if our stomach can digest it and our small intestines can digest it, we can just eat it like food. Like you literally can eat bacteria like food. It's the bad guys that have learned the ways past these barriers, right? And past our defense systems that can still make us sick, right? Because we're studying all this stuff. I was like, wow, this is a pretty awesome system, right? We've got mucus, we can trap it, we can push it where we want it to go, we can put it in our stomach, we can digest it. How is it that we still get sick? Well, there are bad guys out there that know how to get past these systems, right? I had a really awesome dog, Chow Lab Border Collie. He was the best. Nobody came in my yard. Nobody, I'm not kidding you, when he was in my yard. Even I was scared once when I approached and he didn't realize it was me. He scared me. I was like, Blue, it's mommy. Stop. You're terrifying me. Right? So, I had a fenced in yard. I had a terrifying dog, right? Alarm system. Neighbor who semi-retired works from home, right? Was I confident that my house was never going to get broken into? No, if somebody really wanted what I had inside, they'd find a way to get in there. And that's the thing is, for microbes, we are a big fat juicy cheeseburger to them. We are food in home, and they need that. So they're willing to fight for it, right? So some of them have adapted the ability to seriously fight for that juicy cheeseburger. They want you bad, right? So they know how to get past your wall, right? Although the good news as far as skin goes, y'all, there's very few microbes. I think there's like a few worms out there that can actually burrow through your skin. There are no bacteria that can actually cause destruction to your skin and get through your skin. Your skin is probably the most awesome protective barrier you have. Intact skin. Is our skin always free of abrasions and cuts? Unfortunately not, right? But I don't know any of you guys, but unless you're scooting around on your ass, your bum is usually intact, right? You can sit on a public toilet. You do not have to hover over it and pee all over the frickin' seat, right? You can sit your ass down on the toilet seat. Your skin, as I said, is the most protective barrier you have. Unless you're scooting around on your ass and you have a whole bunch of cuts on your bummy, you should be fine to sit your ass down on the toilet. Even a public toilet. Do you understand? Right? Yeah. So, who's the hoverers in here? Quit hovering. You hover too? God damn it. No, y'all just pee all over everything. It's a contest. Oh, yeah, really, you can sit. And I mean, like, if you're super anal, you could wipe it and sit your ass down too. That works too. For the person before you that just had to hover and piss all over the seat. Right? Even if they've got bacteria in that piss, which, unless you have a bacterial infection, your pee is sterile, by the way. you should be fine. So, awesome barrier, right? Protects your ass, literally protects your ass. Then, the mucus itself has antimicrobial substances in it, right? Your tears have a ton of antimicrobial substances. Your saliva, again, has got antimicrobial substances plus enzymes, right? Which they don't care whether it's like a bacteria you just inhaled or, a piece of cracker. They're still going to try and digest it. Enzymes attack molecules. They don't care what molecule, where it came from, right? Whether it was an organism or a plant, right? It's going to eat it. So, okay, the bad news is if skin is not intact, right? Or you've got mucous membranes, but these suckers can swim through it, that mucus, right? Then we've got to be able to detect when that barrier has been breached. Those are our sensory systems, right? And some of them are just like the sensory system to your house, right? The alarm system. If your husband forgets the code and opens the damn door, the alarm is still going to go off, right? Because the door opened. So some of these systems, again, they just detect and they send off the alarm and somebody's going to come, right? In the case of our alarm, hopefully the monitoring system or whatever sends the police or whatever. Or the neighbor calls the police because the alarm next door is going off. So for our body, that alarm, right, is our sensory cells, those phagocytic cells that have those receptors, or those sentinel cells that have those receptors. They're going to send off the alarm. They're going to call in the police, right? More phagocytic cells. Inflammation is going to happen. And some of them can directly destroy the invaders. So in addition to the Toll-like receptors, right, on and within the receptors within our cells, we also have the complement system. And this system in itself can trigger, right, a response, as well as it itself can destroy the invader. See, I like my dog. He would bark and he would warn, but he would also probably freaking eat you if you hop my fence. I kid you not. Because he did eat the UPS boxes. We used to joke and said that, he was a bomb sniffer in a past life or something. All my UPS boxes had teeth marks in them. He punctured his own bed, too. I was like, dude, that was for you. That was your therapeutic bread, you old grumpy goose. So again, too, once they get past that sensory system, right, those security cameras, those detection things, we have the SWAT team that's going to swoop in, right, and take care of the invaders. So that's when we really get serious, right, where we have phagocytic cells will actually destroy it. Complement activation can actually produce proteins that will lyse the cells themselves. We have the inflammatory response to pull in even more phagocytic cells to the area. We have interferon, and the cells are going to respond accordingly for that. And then we also have fever response, right? These are all meant at eliminating the threat that has gained entry into the body. Make sense? Right? And again, it is our security system. So we're going to now go into a little bit more detail, be able to list and define those first-line defenses, including our physical barriers, some of the antimicrobial substances and specifically how they work, and the normal microbiota that you have associated with your body. So your skin, right, multifaceted. You have the epidermis in the very outer layer is actually dead scalp cells that have been keratinized, right? That protein builds up in them. This creates a nice water-resistant coat for the most part. The other good news is any bacteria that land on it, we're continually sloughing off those dead skin cells. And depending on how much dirt you actually track in your house, most of the dust in your house is actually epithelial cells, your own epithelial cells, which is in turn what the dust mites eat. Right? This is what they eat. So it's just transient flora, transient, using us as a ride, right, that you'll find in the epidermis. In the dermis, in the deeper layers, right, is where you'll find normal flora, ones that are normally associated with our skin, right? Like the yeast that we normally have, Candida albicans, that yeast that can overgrow sometimes and cause problems for people if you have too much moisture, right? And if the bacterial population has been changed, if that ecosystem is out of balance. Women experience this, unfortunately, right? You get the whole moisture problem with the vagina. You kind of can't eliminate that. It's always a given, right? But most of the time, your bacteria, your lactobacillus and stuff, keep the yeast in check. It's when those bacteria have been eliminated, maybe because you took too many antibiotics, or you produced too much sugar because you're diabetic. Hormonal changes can screw that whole shit up too, right? So there's lots of factors that can disrupt that ecosystem there. But you have bacteria living all throughout your skin, and they're usually, you know, living symbiotically with the yeasts, the molds that are also associated with your skin. So I wanted to point out, so the most common part of your skin, your dermis, that can get infected or microbes can overgrow, so even just normal ones that are associated with your skin, you can get an overgrowth. We commonly refer to these as zits or pimples, right? And this is where typically what gets infected is the hair follicle. And the reason for that is the sebaceous or oil glands that are associated with the hair follicle. They produce the oily substance that allows the hair to slide out the follicle. Well, sometimes we overproduce that. Again, could be multiple reasons. Could be hormonal, could be diet, it could be genetics, right? That you produce so much that the bacteria are able to overgrow in that hair follicle, right? And then you get the infection, that pus, right? And again, that's dead neutrophils. It means that they're not taking care of the problem. So when you, which is hard to resist, when you pop that zit or that pimple, you're just spreading bacteria into other follicles in the area. Your best option is to put a little bit of triple antibiotic ointment on it. Because again, it's what? A bacterial infection usually of a hair follicle. So treat it as such. Put some triple antibiotic ointment on it. Yeah, good old Neosporin. Yeah, I buy the cheap stuff. Literally just triple antibiotic. But Neosporin is triple antibiotic. Yep. So what's so great about our skin is that dermis layer is really tightly woven. Those cells are really stuck together. There's really not a lot of gaps. So it's really difficult, as I said, for the microbes to penetrate. And then the epidermis is mostly dead, especially the outer layer, and they're filled with keratin, which is somewhat of bad news when it comes to fungi, because there are some fungi that like to eat keratin. They have enzymes, keratinases, that can break it down. But the good news with that is usually it's not available to them unless you have excess moisture. So when you get like jock itch or athlete's foot, right, that's actually, you know, a fungus that is commonly found in the environment. But when you have that moisture, it separates those cells and it makes it more available to the fungi actually being able to digest the keratin in those cells and break it down. Diaper rash, right? That whole problem of sticking a diaper on there creates a warm, moist environment. People who are obese and have the skin folds, right, they have to worry about the moisture that builds up there, right? A lot of them will put things like talcum powder, and if you listen to the radio lately, I guess talcum powder you might want to skip because apparently it could possibly be cancer causing. Great. But you want to keep that dry, right? Because that's that moisture weakens that layer and makes it more susceptible to invasion. So repels water and maintains a dry environment if we keep it dry, right? But if you do things like encase it, you know, in a diaper or shoes that don't breathe and you sweat excessively, then you can run into problems. I have a friend of mine that runs long distance races. They actually make sneakers with ports in the bottom so the sweat can drip out. I was like, really, dude? I was like, yeah, I'm glad I don't need sneakers like that. I was like, that is gross. But you can see the reasoning behind it, right? You don't want that sweat building up in there. It would create problems. My son actually has really sweaty feet. We had to buy him, like, we can't buy him leather sneakers because they don't breathe, right? And they have the, nowadays they have the special dress code in school. You have to wear all black, like literally all black or all white. Like they can't have any logos or, yeah. Which is not easy to find at certain times of the year. So thank God for Amazon and the internet. Because we bought him some nice canvas sneakers that breathe so he wouldn't get athlete's foot again. All right. So these are things, right, that you have to take into consideration. And then if they're too dry, right, they crack and bleed. So you got to find that happy medium. So as I said, digestive system, right, straight shot through, basically, right? Respiratory, not a straight thought through, right? So we have to have that protection. We have those ciliated cells that push that mucus up and dump it into the digestive system so we can push it out the body. Urogenital, again, mucus-bathed, ciliated, move that stuff out. Keep that stuff out. And then the best thing you can do to avoid urinary tract infections, anyone know? How do you keep stuff from going up the urethra, taking up residence in the bladder, even worse, traveling up the ureters to the kidneys and causing problems? Right here, baby. Drink lots of water. Because what do you do when you drink lots of water? You gotta pee. So that means anything in the bladder goes out. If you continue to flush the system, then something can't take up residence, right? It's when you leave it in there, right, for long periods of time, then it's more apt to take up residence. Drink lots of water. Pee often. And he's like, yep, I got that down. But that's the other thing too, you gotta pee. You can't hold it in, right? Water can be toxic if you don't pee, right? You can die from water toxicity. Some woman trying to win like an Xbox or something on a radio station several years back literally killed herself. Yep, it was pretty stupid. Shame on them. So how are the microbes trapped in the mucus removed? So for the digestive system, right, we're continually, unless something goes wrong, right, that system is always pushing stuff out, right? Constant peristalsis, contraction of the muscles of the digestive system, we're pushing it out. Respiratory is that mucosal escalator, right? You get the mucus produced, but then the ciliated cells push it up, right, and then it goes down your esophagus and then expelled through the digestive system. You're actually meant to swallow your spit. People who spit, depending on the mood I'm in, will sometimes get a lecture from me. Also depending on whether that person might shoot me. Nowadays I don't usually say anything. I just make faces and grunt. But think about this. If you're producing excess mucus so much so that you feel you need to spit it out of your body, if your body is producing more mucus than normal, what could possibly be in that mucus? Bacteria, viruses. What did you just do by spitting? Exposed others to your yuck. So in this case, yeah, your cooties, right? Spitting is the worst thing you can do. People who hock a loogie on the sidewalk. I'm like, really? Thank you so much. You just contaminated all that air, and then as that thing dries up and people kick it, it just goes into the air, right? And if you have something like tuberculosis, guess how many bacteria you would have to inhale from their loogie? 10. 10. What's the chances that 10 might be in that spit? Oh yeah, pretty high. Pretty high. Right? It is the most disgusting thing that humans do. Swallow that shit. Or spit it someplace where someone else hasn't got to come in contact with it. Spit it into a napkin and throw it in the trash can. Right? Be considerate of others when it comes to your crud. Right? You wouldn't blow your nose on somebody. Right? When you sneeze, what should you do?
I hate when people sneeze and then I can smell the sneeze.
Yeah, if you smelt it, you were too close and you inhaled it too, right? And now you're really pissed.
Yeah.
Okay, this is what they teach at my son's school. Vampire.
It smells good.
Right? You sneeze into your elbow. Why don't you want to sneeze into your hands? Because you touch everything with your hands, right? So then you're going to go contaminate a doorknob or, the keyboard or whatever. You want to sneeze into your elbow.
It's a real thing, I promise.
I believe it. She is a very sensitive sniffer. No, but yeah, and when you talk, right? You laugh, right? Yeah, well, face talkers, right? We've all experienced this. You know what I'm talking about, right? Those people that have to talk in your face. Don't back up. They keep coming, by the way. This is how you protect yourself. You put your arms up, right? Arm's length away is about a safe zone, right? For that whole respiratory droplet problem, right? Of them talking and spitting on you, right? Some of them are really, you might need to stick a leg in their face or something. But yeah, usually the arms work, right? If they don't get the clue, they at least, you've created a barrier, right? That hopefully they Red Rover, send Sally right over. We won't be breaking down that barrier. Unless they're drinking. And in that case, then you just leave.
Or it's another culture, like some cultures, it's rude to be further away. You're supposed to be close enough to smell their breath. Like,
Yeah, no. I'm good. I'll pass. I'll be handing them a breath mint. I actually don't have, usually, not until I figured out what was wrong with me. I usually didn't smell most of the time, but now I do smell. And sometimes, like you, I'm like, oh, God help us, right? God help us. So, let's see, what do we got left in this? Yeah, we might need to just take a break, huh? Yeah, let's take a 15-minute break. Okay.